in general.
The cellular.
Protein coats, some of them have a membrane that makes them, like capsid or hallucinated.
Okay, so all of them will have a capsid, a protein capsid. But it's the envelope that some may have and others do not have. And those are the ones that are called naked viruses. So I believe we left off here. So this is specialized transduction because when the bacteriophage introduces its genome into the bacterial cell host, the genome will insert itself on the genome of the bacterial cell. And so when this happens, then sometimes because of some kind of stimuli, the genome of the viral genome will excise from the viral genome will excise from the bacterial genome, and with it now we'll have a piece of the host genome with it, as you can see here. So here comes just the DNA, the viral DNA or nucleic acid, and then you have the incorporation here or insertion into the host genome, and as it detaches from that host genome, then it sort of takes a piece of it, and so now all of these new viral particles are going to contain part of that genome from the host. So this is one way on how things could be carried from one cell to the other, as those host cells not only, well, not in this case, the bacteriophages, they will carry part of that genome from that host cell into other cells and will transfer that per se mutation into other bacterial cells. And this is another bacterial cell that is being infected by this bacteriophage that is containing both the viral and the host genome, the previous host genome, into the genome of that host. And so it inserts itself here, and again, that's how these mutations happen. So that's specialized transduction, which we looked at in chapter 11, remember, transduction? Okay, so that's for those complex viruses. That's how we call bacteriophages, right? Complex viruses. Now, these are animal viruses. Why do we call them? Why is it different from here? Tell me the difference between here and here. I mean, there are a lot of things that are different, but one thing that sort of pops out from the whole cells.
The nucleus.
A nucleus, right. So this happens in animal cells. And what we have is the replication of viral particles inside the nucleus of that host cell. So again, the first contact of the virus with the animal cell requires for the receptor specificity between the two. So there are receptors on the surface of these host cells that are very compatible with the receptors on that influenza virus. As they come together, then the virus envelope will fuse with the envelope or with the plasma membrane of the cell. That causes for the host cell to membrane to go through ruffling, so it starts doing something like this, forming a vesicle, and it brings that virus inside. And that's all triggered by the interaction of the receptors between the virus and the host receptors on the membrane. So once inside, you have the uncoding of the viral genome. The viral genome then travels to inside the nucleus of that animal cell, where they will not only they carry the genome, but also an enzyme that will help them do the replication of the viral particles using, of course, the machinery of the host cell. And so, but the replication itself happens here, or the transcription of that code or that message happens in the nucleus, and then you have the messenger RNA taking some of these information to make new viral proteins into the cytoplasm, and here you have the rest of the process of viral replication, which is very similar to what happens here, which is biosynthesis, all of the different proteins being made using the ribosomes of the host. You have maturation and you have lysis. This is different. I just wanted to show you that this process is basically the same when it comes to making those proteins. And so here you have biosynthesis, and then after this picture is not showing it. You have maturation, so all the viral particles come together, making the viruses, and then the viruses will bud off from the animal cell. Yeah.
Okay, so in the phages, when they're replicating, the cell lyses and splits open.
In the lytic cycle,
and then-
in the lysogenic
they
cycle,
don't.
right.
But they, eventually, when they do, the lysis cycle, they do split open. That never happens for animal cells?
It could happen, but usually this is what they do. This is a more normal process in animal cells where they bud off. I think I may have a picture here. So here you have the virus making contact. You see the ruffling of the membrane here. This is an electron micrograph of a viral particle or viral particles trying to get in. You see how it is enrolled here. And in the same way, I don't have a picture of that. But in the same way, it sort of buds off from the cell, from the host cell. One thing that happens here is that when they enter here, some of these, they will leave some of those receptor particles, they will leave them on the surface of the cell. And so as they come out, they pick up some of those that belong to the virus, right? And we'll see something that is more, that illustrates this process very well when we look at the HIV virus. So
Thank you.
as they come out, again, they pick that up and they bud off from the host cell and they go and infect other cells. Now, here's what happens with influenza every year. We get one form of the virus that may have many different combinations, a recombination of different variants. And so you have, for instance, two different strains here, having not only a different genome, but it has different receptors on the surface, right? So as it combines, like during the year, spring, summer, as it combines, so if you have this recombination, then what we get in the next fall is something that is a mixture of many different of these variants that the virus have been in contact with. It could be an animal, avian, or porcine, or human even. So is one of the reasons why we get a vaccine every year, because this happens every year, and we don't know. By the time we get the virus or somebody gets sick, we don't know all of the variants that is contained in that, all of the different genes that is contained in that new variant. And so we get the vaccine that protects us somewhat, but not 100%, because we're not aware, or we don't know exactly the composition of that new variant. And so as it recombines, then the combination of these two, look, now it has not only the genome, the genes of these two, but also the receptors are a combination of the two previous variants, and maybe even others. So so that's difficult to sort of treat right away, but with the new technologies, it's possible maybe one day to get the vaccine within enough time to treat people maybe within a month or something, but it's too early for that. We're still getting, we still don't have 100% protection with the vaccines that we get. And how do we know that? Well, look, this is the 1918 pandemic. This is the variant, right? The H1N1. And the pandemic in 2009 is basically a combination of not only other variants, the Eurasian swine, the human, different variants, H3 and 2, but it has part of the avian gene pool, and it even has genes that composes this variant from that caused the 1918 pandemic. And we're able to see that, detect that with these molecular methods of identification, looking at the genes. So this one, you see the gene here is basically what we have here. And that has been passed on to previous strains or variants that have caused disease in different years around the world. But so it's sort of like a roulette kind of thing that you have, all of these things combining.
What does antigenic mean? Like it's coming from a before, like an earlier ancestor or something?
Antigen is a molecule.
Oh, the antigen.
Yeah.
Ah, okay.
Antigen.
That's what that refers to.
Yeah, it's a molecule that is present. Even our blood have antigens,
Yeah.
right? But in this case, these are molecules that are foreign to us, and that their nature is viral, right? So yeah, you get this recombination, and it's something that these guys are ahead of us. They're always like changing their mutating process is very rapid because of their life cycle. How many variants do we have of COVID-19, of the coronavirus, since was detected in 2019?
Over a dozen?
I think we went over
Oh,
that.
okay.
So many variants. And it's within a period of what, 2019 to 2025, that's 6 years. But the amount, I think later on, I have something, I don't remember if it was in this lecture or another one where we have, I think it's in epidemiology, that I have a sort of a map of how many different ones have arisen since then. So RNA viruses, so we have DNA and RNA viruses as mentioned. And one of the things about RNA viruses is that they're not like complete to replicate it right away, especially if there's a negative strand RNA virus. They cannot replicate right away when they infect a host. The positive single strand or a positive sense RNA molecule is able to replicate its nucleic acid. But if a virus contains a negative one, brings the negative one, and that negative strand is used as a template to produce a positive one, okay? To complement it and produce a positive one. And then that positive one now is able to start the replication process. But a negative strand alone is not able to replicate. It has to be converted first to a positive one. Okay, so that's one of the sort of natures of the nature of this particular type of nucleic acid. The DNA is no problem, but an RNA one, and you have a positive and you have a negative, the positive can replicate, but the negative cannot. All right, so again, here's the process of replication of viruses in animals. Attachment, penetration by endocytosis. What is endocytosis?
Brings it in.
Engulfing, right. So it's sort of like eating, bringing something inside the cell, or fusion. Again, the process is the same, where you have the encoding of the virus, of the envelope, the release of maybe one enzyme that they're bringing, and usually it's just one enzyme. But you're going to see with the HIV virus that there's more than one enzyme coming in, or accompany that nucleic acid. Then you have the biosynthesis, producing the nucleic acid and the viral proteins to make more viral particles so that they can continue going to infect other cells. And maturation, where the nucleic acid and capsid proteins are assembled. Again, this is one of the processes that I'll show you how it's done. It's one of the last things. The capsid is the one that protects the nucleic acid. So sometimes the proteins are made, but they had to be sort of like finishing during assembly, finishing like one last step before the entire virus is put together. And then again, released by budding or rupture. So they could, like the HIV virus is one that could rupture the cells. And when they rupture, it's because they have so many viral particles inside, and they just open up, release all of them, and this is how our T helper cells are destroyed. So you have less T helper cells being active or being available to help in the attacking of these viruses. Because they're being destroyed. All right, so a little bit about influenza, a little bit more. This is as of from 2000 2020 or 2019 through 2025, you can see that these peaks are very low here. Do you remember that? During the years of the pandemic?
Oh yeah, COVID. I mean, the flu was low. Like it disappeared. They said
Yeah, it almost disappeared. And why?
because everyone had COVID already. I don't know.
So you think it's because everybody had COVID?
No,
Anybody else?
probably not.
Wanna take a guess?
Oh, I was saying that many of the people who were susceptible to COVID, people who were already sick and were going vulnerable to death from a respiratory virus, they would, COVID has a higher baseline mortality than influenza. Influenza one in, I don't know, 100,000 was COVID. In Italy when they had COVID, it was 3% or 4% of people were dying from COVID. So all these people getting COVID, they were dying from it, and they were dying from COVID before they could get influenza.
And they
Yeah.
stayed inside too, I guess. People.
Yeah, so all of these factors, one very simple and important factor that helped in having this number so low was, staying inside, isolated, not, in too much activity with others, getting together with others, but also wearing that mask.
Oh yeah.
Remember wearing the mask? Wearing that mask help in this virus from passing from one person to another. Pre-COVID pandemic, we used to have about 20,000 deaths, not just infection, but deaths in the United States. That probably is sort of getting back to that. I mean, I think one thing that we learned about with the pandemic was to use that mask. I see people wearing that when they have, like maybe the flu or some kind of respiratory issue, they wear the mask because it does prevent from these things from spreading, from being passed from one person to another. But after the pandemic and after we went back to working and going to school and to our normal activities, look at those numbers go back up. I think I have the numbers exactly for which you're studying the season. But there are other types of viruses that have been sort of silent at times, but they have caused a lot of deaths, a lot of grief in many parts of the world. And this is one of the most, deadly virus that we have had in Ebola. And we had a few cases in 2022. You get a case here and there in some of the countries in Africa, but one of the really bad epidemics, because it wasn't a pandemic, was the 2014 epidemic in East Africa, West Africa. And that caused for the death of about 11,000 people. In people in these regions of the world, they're kind of used to dealing with some of these diseases. But still, a lot of people die because there are some cultural sort of rituals that they follow to bury their death. And so before they could get to all of this PPE or personal protective equipment, maybe they don't know. They're not aware that the person is infected with Ebola. They start cleaning them and, this is transferring to them and this is how the disease spread. So we did have a lot of deaths in the thousands, I think around 11,000 in these countries of West Africa. Here's Ebola. It's a spiral type of virus. It's pretty large, close to 1 micrometer in diameter. These are some of the areas where it is endemic to. And usually it appears when humans have some kind of contact with wildlife from the forest. The symptoms are very similar to any type of, disease. Headache, fatigue, fever, muscle soreness. What am I talking about? Am I talking about the flu or, right? A lot of the common infections that we deal with, they have very similar symptoms. But then you start having other more serious things or symptoms, such as having vomiting blood, you're more fatigued, probably losing a lot of the blood, a lot of the oxygen, and you get bruising, and all your organs start sort of like bleeding, you start bleeding inside, and eventually that internal bleeding and damage to all of these organs causes death. And see, here are some of the, as of 2022, the government of Uganda declared that there were some cases there. The thing about, let me see, I have it here. Okay, so
Is it spreading in the first 7 days before they get the symptoms, or is it just spreading after that?
well, it's in the last days or when the people start feeling the symptoms,
That's when it is.
yeah, start spreading. And I think it has an incubation period of, if I'm not mistaken, about 14 to 21 days.
Yeah.
We only have one fatality in here of a person that traveled to one of these countries, and the person got sick there, and he was brought back to the state in a sort of like a encapsulated kind
A
of
bubble.
a machine by the Air Force. So something that was completely isolated from the rest, so that it wouldn't transmit the disease to others. That person died in Texas.
Yeah.
And we had a couple more people, one
The
guy
doctor.
in New York, and
Yeah.
a doctor who was riding the subway.
Yeah, that's what I was thinking. I remember people freaked out because he like went to Brooklyn or something, and then he came back, but he was already feeling sick.
And when he realized that he was like that, then
Yeah.
he quarantined. He also had I think it was a nurse from Vermont or something that also seemed to have been positive, but she recuperated, she got well, and the doctor as well.
Yeah.
So it was only that guy.
Yeah, my so I'm a paramedic, so this is before. So the unit that I was working was like a hazmat unit. So my old partner, they had to go pick him up. So like afterwards they took him to Bellevue, and they had to do. So the doctor, he called 911 himself and said, I think I have Ebola. So they knew going into it and at the time Ebola was going on, especially in New York City. Like the fire department was preparing for it. But he called ahead and said, I think I have it. So they went in with like all their hazardous materials protection, and they wrapped him up and brought him in the ambulance. But the crew that was with him in the ambulance had to be like fully deconned at Bellevue. But Bellevue is prepared. They have a corridor, so that it's like perfectly made for it. They're very prepared. For one, I mean, if it had been like a city's worth of Ebola, maybe not. But, yeah.
Well, you remember when the COVID-19 pandemic happened? We were running out of places in the hospitals, and that's why they had to put that
in the park.
In the Jacob Javits and that boat by.
Oh yeah.
Crazy. But yeah, this is even worse because the dangerous level, the biosafety lab level is a 4 for this. Biosafety level 3 will be for COVID. So it is transmitted through, contact and stuff like that, but it's not like through, and probably through aerosols, but not like Ebola. Ebola is really, you really have to be suited up, no contact with the air, for those that are dealing with the patients. And we'll go back to this when we do epidemiology, because a lot of the countries that are endemic to this disease, they are, sort of kind of prepared and not, because they don't have the resources that, like these, in case rooms and things to build, like Bellevue, to deal with these things. I was gonna show something here, but, let's see. Yeah, this is still 2022. Okay, but one of the things that happens with Ebola is something very peculiar. It hides. You get an outbreak, some people die, and then all of a sudden, boom, it disappears. And so no more cases. So you think that, oh, okay, that's it. Well, it hides in the testes, it hides in the cornea, in places that are called privileged places, where the immune system is not gonna go and try and find it and get rid of it. But and then they hide there, and then some kind of stimuli will reactivate them again, and then you get a couple of cases. But according to, when you look at epidemiological data, you see that it emerged in the 1970s. And you have sporadic cases of Ebola, a couple of cases here, 5 there, in different countries in the tropics. But they disappear, but it's always constant in the population. Okay, it is endemic to these areas, and it doesn't disappear completely. Let me just go back to, yeah, this is 2022. Perfect. Okay, so here is HIV, and this is called a retrovirus. Anybody took, what's the class? There's a class that you guys need to take for nursing or for occupational therapy. Anyway, it's where you go over this. Have you? Anybody gone through to study HIV in detail?
No, it's paramedics who have to do that, HIV in Society.
Right.
But that's paramedics.
Okay.
Yeah.
Here or somewhere else?
Yeah. But
Okay.
I don't know anybody who's done that.
Yeah, I know there's human, something. There's the health sciences have a course that is just AIDS in New York, something like that. But anyway, so let's go over this. So this is a virus called retrovirus. Why? Because it comes with an viral RNA molecule, right? And then it uses that to make a DNA molecule. So here's what happens. We have the virus, right? This is a T helper cell, which is specific to this particular type of virus. HIV virus has these receptors, GP120, that attaches to the CD4 receptor on the T helper cell. This is the CD4 receptor on the T helper cell. So remember, the specificity for any type of virus and the cell that it attaches to. So those receptors, they need to match. These two match. However, there has to be another receptor involved, which is the CCR5. This is the one, remember, I spoke about it, the mutation that some Eurasian groups have. Well, when there's no mutation and this receptor is normal, it helps in the final configuration, the final attachment of the virus with the T helper cell. So virus comes, GP120 attaches to the CD4, then the CCR5 helps in configuring the two other receptors with it. And this way, the envelope of the virus will fuse with the membrane of this cell, of the T helper cell, which is part of your immune system. And then as it fuses, then like we saw in one of the previous slides, is an animal virus, right? So it sort of ruffles the membrane. This will fuse and will penetrate the capsid will penetrate, and together with its DNA, with its RNA, it will bring 3 different types of enzymes with it. So it brings transcriptase to go through the process of transcription, integrase that will cut the genome of the T helper cell and will insert itself there. Then the final type of protein is protease, and the protease will help sort of with the last processing of the proteins being assembled for the final viral particles. So let's go over this process. So here is the viral RNA with the 3 different enzymes. It comes, remember, RNA will serve as a template to make a single-stranded DNA. That single-stranded DNA, because it was made from RNA, right? It sort of has mistakes. It's not completely normal or good. So now it will use that DNA, single DNA molecule to make a double-stranded DNA. So now you have the double-stranded DNA. The copying of that RNA into DNA is done again by reverse transcriptase. And then once you have the double-stranded DNA, it goes through the nuclear pore of the DNA molecule, of the T helper cell. So it goes through one of the pores. It takes that integrase enzyme and it cuts in certain part of the DNA of the animal cell or the T helper cell, and it inserts itself there, right? So now the genome of that T helper cell has the viral genome contained in it. And so if this cell was to replicate before it gets destroyed, then it's going to produce or carry that viral genome in there. So now, after and this is called the provirus, when it's inserted into that genome of the T helper cell. So now you have the copying of this code that is for the different proteins of the virus is being transcribed. Remember transcription? It's being transcribed into messenger RNA. That messenger RNA is going to go into the cytoplasm, and using the ribosomes of these T helper cells is going to make the rest of the molecules that make up the virus. So once all of those parts are ready, then you have the assembly, right? The assembly and the maturation. So now you're putting together all of the parts of the new viral particles, and you get new virions, okay? And the new virions are going to go into close to the plasma membrane, and there they're going to bud off. When they bud off, they pick up some of those receptor proteins that they left as they enter, and they detach. So they bud off from the T helper cell. Now, the T helper cell now is left with maybe infected with some other particles, so it could be lysed, or it could continue replicating with that provider a molecule inserted in the host DNA. And then the mature virion will become mature once the last enzyme that the virus brought in acts on the proteins that were produced, and then it sort of rearranges all of these proteins and it cuts them in different parts. I'm gonna show you a little video about that. And then the virion is complete, is mature, and it goes and starts, infecting other cells. So it is called a retrovirus because it uses reverse transcriptase to make DNA from RNA. And we had an epidemic. We still have a lot of cases. They are still in New York, San Francisco. People don't die of this much anymore because there are medications that people take. People may be more aware and more careful, and do not get as infected as the time when we didn't know much about it. But we still have a high incidence of the virus or the infections in certain parts of Africa, and that seems to correlate with the incidence of certain types of cancer in those areas. So it's still a problem because people tend to forget that these things could kill you, right? There is a movie, very old, 1993, that talks about the biology, the epidemiology, how the virus started spreading in New York and other areas. And it also includes the political, socioeconomic aspects of the virus and why we reach such high levels of mortality, because there was not enough resources put in by the governments to deal with the disease. Anyway, so that's what we have with HIV. It's not the worst thing right now again because some people take medications if they think they have been exposed. But when you don't have resources, like in low socioeconomic countries where there are no resources, then people still die from this stuff.
So, how does the retroviral medications work? Like when you know you have it, so you take them like monthly or however often, do they stop this process?
They can stop different things. They can stop the attachment, the fusion. They can stop this process here of making DNA from RNA. So it's a cocktail. So you take different drugs. You take like 15 types of pills.
Wow.
And they all target something. And we're gonna look at this when we do antimicrobials. We'll look at antiretroviral therapy, which includes all of these things that, prevent some of, so if one thing didn't stop it, maybe it stopped it, in another way. So I'll show you the very nice movie, little clip that shows the viral replication of HIV. First, and this is embedded there, so you can watch it, later.
So this is HIV, it's a typical retrovirus, meaning that it has an outer envelope, and in the center it has 2 copies of RNA, as well as an enzyme here in blue that's reverse transcriptase, which will ultimately turn that RNA into DNA. The virus itself, with this outer envelope protein, actually directly infects T helper cells. The way that it does this is that as it comes up to the cell surface, it uses receptors that are on T helper cells and exclusive to T helper cells, which are CD4 molecule, which really defines T helper cells. It's a surface receptor that binds to the envelope protein. That causes a conformational change and allows a second receptor to grab hold of the envelope. This is the chemokine co-receptor. It's also called CCR5, and we'll talk about that more. What happens now is that the stalk of the envelope protein pierces through from the virus into the host cell and starts to draw the two cell membrane and the viral membrane together. And what ultimately happens is fusion of those 2 membranes, and the viral genetic material is injected essentially into the cell, and the envelope protein is left at the cell surface. The virus has a matrix and a capsid protein shown here in green and red that essentially are digested when it enters into the cell. That releases viral enzymes and the viral RNA. And here we have reverse transcriptase, which takes the viral RNA and using host nucleotides converts that viral RNA into a single strand of DNA. While it does that, it makes some random errors, which is characteristic of reverse transcriptase. It has very poor proofreading activity. That single-stranded DNA now is again reverse transcribed into a double-stranded DNA. At that point, another enzyme that has come in with the virus in the beginning, called integrase, essentially grabs hold of that double-stranded DNA and carries it through a nuclear pore into the nucleus of the cell. Within the nucleus of the cell, it finds the host chromosome, and it basically, the integrase enzyme makes a nick in the host DNA and allows for HIV to insert itself into the host chromosome. And that right there is what establishes lifelong infection. Now, RNA polymerase comes along and makes messenger RNA. Those messenger RNAs encode for different viral proteins. They end up associating with ribosomes on at the surface of the rough endoplasmic reticulum. And here's a piece of mRNA that's making envelope protein, which is directly produced into the endoplasmic reticulum. And it's shoved then through the endoplasmic reticulum and taken to the cell surface, where at the cell surface it becomes embedded in the cellular membrane, and at this point coalescing with other envelope proteins that have been produced. You have this cluster of envelope proteins now on the surface of this infected cell. At the same time, there are other messenger RNAs that are being produced that allow for translation of other viral proteins. So here are additional viral proteins being made, which are going to be used to make up the key components that the virus ultimately is going to need. These are transported again to the cell surface, to the area where these envelope proteins are, and a strand of RNA as well as some of the enzymes are part of that complex. This then buds off at the cell surface at this point, but it's still not a mature virion because the polyprotein chain needs to still be digested into its component parts. That's done by an enzyme called protease. Protease breaks up those polyprotein chains and ultimately allows for them to coalesce and form the mature structures that make up the final virion. And now you have a mature infectious virion that can go on now to infect other cells. Once that happens now, the cell can produce tons of viruses, and this is really what then keeps the whole process going.
So, it's very different to what happens with other type of viruses and how they replicate, so it's very unique. Alright, so other viruses that we may deal with, and may have dealt with for a long time, is measles. And, it reemerged in 2019, and in 2024-25, we have a high incidence of measles cases in Texas, and a few children died. So it is a virus that could be lethal if and it could be prevented from causing disease and death just by simply vaccinating. And that's the case for a lot of the viruses that are very deadly. There's only one way that you can, because of their a high rate of mutations, the only way to prevent them is by vaccinating, because it's very hard to deal with them, and they are, they're antigenic, not knowing all of the antigen variations that they have. So that may be one way to deal, or the best way to deal with these viruses. So this starts as also like a regular type of, or your average type of virus, rhinovirus, where there's cough, there's discharge of the nasal, from the nasal cavity, you have fever and stuff. But the disease progresses, and there are these very specific signs, such as having these spots in the back of the mouth. They're called Koplik spots. There's also a rash that develops. And again, we have these outbreaks, these darker blue areas is where we have most of the cases. And they tend to be correlated with areas where people do not tend to vaccinate their children. Here's what we call a persistent infection, and this is caused by a virus that it doesn't really disappear. You may get better with treatment and stuff, but this is one that if you have been infected with the varicella zoster virus when you were little, you are prone to get, so if you had chicken pox, you could develop at certain age shingles, and if you never had chicken pox, then you're okay. But if you had it, the virus remains in your body, and it hides in the spinal cord, and so and it's usually with age that your immune system may not be as strong, and types of conditions may trigger for the virus to reappear. And so you have this rash that is traveling from the back of your body into the front, and it seems to be very painful because, again, now is in the nervous system. So it's almost like burning, like a burning sensation, or at least that's what people that have had it explain the feeling that they had. Luckily there is a vaccine for this. You can only get it when you're older, like after 50, 55, you can get the vaccine, but it's one way of preventing having something that is very painful and probably, you're not able to go to work or whatever, so
Do you only get the shingles vaccine if you've had chickenpox?
usually, yeah.
Right? Like, why would you get it?
No, you get it if you had it.
Yeah.
If you didn't have chickenpox when you were little, and I know people that have it, people that maybe come from an island or something, never had chickenpox, then they,
And you
no. All right, and COVID, we know a lot about COVID, right? So many dead, and also, so these numbers are kind of old. So this is the number of people that died as of June 2025 around the world, and then in the United States, 1.2 million. Again, because it has become an endemic type of virus, and we know more about it, and we invested not only in the vaccines, but also in prophylactics treatment for people that do get infected. We kind of decrease the number of not only cases, but the number of deaths especially. There's some links in some of these slides that you can visit. Most of the dashboards that we had during the COVID pandemic are not there anymore, they're not active, but some of them are. I think universities and stuff and the World Health Organization, they still keep one of the numbers because there's still cases around.
John Hopkins had a dashboard, except they stopped updating it about a year ago.
Yeah, a lot of places stop. And I guess the danger is not so much because, again, we have the vaccines, preventive measures and treatment for it. And until the next one comes, who knows? This is as of April 2025, this is for vaccines. 2023, you can see that most people, or people that got vaccines, was more right after, during the pandemic, and as people got vaccinated, they also got better, recuperated, recovered from being infected. We also reached this herd immunity, and herd immunity is sort of like a sign that perhaps we don't need to monitor it as much as we used to. You can see that the number of vaccines also has decreased, especially in certain states. And that social media and all of these conspiracy theories have played a role in the decrease of the number of people getting vaccinated. I think, most of you that are studying something related to healthcare, whether it's in animals or humans, you want to know why, or at least the facts, and so hopefully you'll spread that to other people. Monkeypox. You remember monkeypox in the pandemic? It was very, we thought that it was going to reach also very these large proportions, but no. These were more or less the number of cases and number of deaths throughout the world and in the US. And luckily for us, we had a vaccine for it. So here are the numbers of, this is in 2022. I remember as a student that was like, oh, my mom is insisting that I go and get this monkeypox vaccine, and he did went and get it. But these are some of the signs that you have it, and these are the confirmed cases by the World Health Organization. So we were lucky again that it didn't reach those large proportions. Are you guys okay, or you want to take